summer research experience. He worked on a project using zebrafish, studying the toxic effects of neurons of the chemotherapy of drug cisplatin. And later, as a graduate student, he focused on the development of and testing of neuroprotective compound P7C3 in mouse, in rats, in C. elegans, and other cellular models. So now Dr. De Jesus is working in the lab of Dr. Baer to learn and apply in vivo electrophysiology in the context of experience-dependent plasticity. And his goal is to use a theory-based approach in order to identify interventions which promote synaptic plasticity and recovery from neurodegenerative disorders. As we all know, after Dr. De Jesus' presentation, we'll have a time for question and answers period, followed by, of course, the participation of Dr. Mandana Sansafar, who is going to be talking about the different training possibilities at MIT. So without any more introduction, Héctor, always a pleasure to see you, and we look forward to your talk. Thank you.
Thank you very much, Dr. Maldonado. It is a pleasure for me, and thank you for the invitation to you and Marimar Velázquez Vargas for organizing this seminar. I've seen all the different speakers, and it is an amazing panel of people that you have put together. So I'm just humbled and happy to be able to speak to you a little bit of my work in the Baer Lab in the past 4 or 5 years that I've been doing. So the title is Reopening of Juvenile-like Plasticity Using Digital and Pharmacological Therapeutics in Adult Mice. It sounds like a lot of jargon, but what I'm going to try to do and convince you is that we can use different ways, both drugs, which is pharmacology, or sensory stimulation, to try to manipulate the brain in a way to allow us to help strengthen connections in the future memory for humans. So I always like to start by saying that my journey, like you mentioned, didn't start here. I was an alumni at the University of Puerto Rico in Riviera. I was fortunate enough to be in a lab that allowed me to explore many of the things and the beauties of the brain. As you mentioned, I used the animal model of a mouse to study proteomic changes. And that really inspired me to go ahead and do my PhD at the UT Southwestern Medical Center in Dallas. And my passion really is to find treatments, to use what's called translational neuroscience. And to do this, what I've been trying to do is to use different experiences from the University of Puerto Rico, from UT Southwestern. Later, I went to the University of Iowa, where my PI moved in the middle of my PhD. And then now at MIT, to later open up my lab and, like you mentioned, study neuropsychiatric disorders at different levels, from behavior, physiology, circuits, cells, proteins, DNA. I'm a big picture person, so I like to try to understand everything from different perspectives, even though I cannot go very deep, but that's the way I like to do that. And I encourage everyone, to always find the way that you learn and apply things and do it your way. Many people go in and study very specific things, and that's a good way of doing things, but I like to go around, and that's why for my postdoc, I did something totally different from my PhD and my undergrad. So I always like to throw this slide because to encourage everyone to reach out to me or to your mentors about opportunities at different places, like you're going to see today at MIT, and understand that what I'm going to show to you today is a snapshot of something and a journey that I have traveled throughout many years since 2009, I guess. Wow, that's a long time. So again, translational neuroscience. What I like to do is to use mouse models, cells, to ultimately impact the human brain. So it goes back and forth. People think that we discover everything in cells or we discover everything in humans, but in reality, it's more complex than that. We actually find things in humans, try to translate them into mice, to then find therapies to go back to humans. Sometimes we have to go back to cells. And it's very, very dynamic, this process. But in all of them, we can find neurons. Neurons, as you probably have heard from other speakers, are these cells in the brain that give rise to many of our functions as animals, including learning and memory. They're also in charge of forgetting. And I added this earlier today because I was saying probably there's a lot of people that want to forget a lot of things about 2020, like COVID-19. So the brain has this capacity of having these cells or neurons that allow us to do that, to learn, memorize, and also forget sometimes. And the way they do this is by communication. They communicate with each other through what's called the synapse. So the synapse, it's how neurons are connected. They're not physically connected, most of them. They are actually, this is a neuron, the presynaptic neuron, and this is a postsynaptic neuron. And if they want to communicate, what they do is they use electrical and chemical stimulation to do so. These are neurotransmitters, so these are chemicals. And what you're going to see here in this diagram is that when electricity is stimulating the neuron, the presynaptic terminal, that induces a cascade of events that allows for information to then pass from the presynaptic cell to the postsynaptic cell that can receive that information and do something about it. So I always like to show this because, you know, when I talk to most people around that are not neuroscientists, they think, you know, neurons are connected, touching each other, but in reality, they're not. They use this very nice and elegant way of communication called information transfer to try to take the message from one cell to another. And this happens from many millions or billions of cells at every millisecond of your life. So this is happening all the time. But as Carmen mentioned in the beginning, neurons, I study plasticity. So plasticity is the ability of neurons to strengthen or weaken this connection. So as you can see in this diagram, for example, we have a presynaptic cell or neuron and a postsynaptic one. They're communicating. If experience happens, so meaning like they want to fire together, they want to learn something together, they want to communicate with each other better. What they do is they strengthen their connection through different mechanisms. Like you can see here, for example, they put more receptors in the cell surface of the synapse. And now the communication given by this yellow arrow, it's much stronger. There's a term called their neurons fire together, they're wired together, fire together. So when the communication is very good, anytime that the presynaptic cell want to communicate with the neighbor, they can do it. Now, this process again is not one way; it's a two-way street, and sometimes the system or some toxicity in the environment makes neurons to weaken. So the process of weakening is called long-term depression most of the time, and it's just you can take a very good communication pattern and weaken it and make it not so good. In this case, when the blue cell want to communicate with the green one, sometimes they cannot. So it has to reestablish that type of connection. And that's what's called plasticity. So now you understand one of the concepts that I study is this homeostatic process by which you can strengthen or weaken connections in the brain. Now, how do I study plasticity? In my lab, we use the mouse visual system. And I'm gonna give you just a little bit of architecture of the mouse and most animals' visual cortex, where you can see here the eyes where we receive the information from the outside world. It crosses the optic chiasm, the information that goes here in the nasal part of your eye, of the retina, goes and crosses to the other side through the optic chiasm. It goes to the thalamus, where you can find the dorsolateral geniculate nucleus, or LGN, and it goes to the visual cortex and other regions like superior colliculus here. And you can find in the cortex, in the occipital lobe or the back of your brain, the monocular region and the binocular region, meaning that it receives information from one eye or both eyes here in yellow. So here's how the visual cortex of a human looks like. Again, it's in the back. You can see the eyes. optic chiasm, thalamus, and the visual cortex. In a mouse, just to show you some similarities, it is found also in the back of the brain, in the occipital lobe, and you can see here it's V1, and you can see also that it crosses in the optic chiasm. So the way I study activity or plasticity in the brain is by leveraging the concept that I mentioned you before, that neurons use electricity to communicate. So we can then implant electrodes in the back of the brain, in the visual cortex, and record that activity through these LFP electrodes, or local field potential electrodes. So you can see here, it's a surgery where I'm implanting a very tiny wire in the visual cortex that later the animal is going to recover, and it's going to give me access, direct access to the activity in the visual cortex. How this activity looks like, something like this. On the y-axis, we have the voltage. On the x-axis, we have time. And you can see the fluctuations in voltages. This is just an animal looking at a gray screen, so there's no stimulation. This is normal activity. As I told you, neurons are communicating all the time. And we can take, this is the wire that I put in. It's here. It's taking all the information from all the neurons around it. That's why it's called local field potential. And we can take that information, the raw information, and apply some statistical and computational methods, called here low pulse filters, to average the response of millions or sometimes thousands of neurons around the area of the electrode. And that's what's called local field potential that I'm going to be showing you later. Now, how does the animal look like? This is how the surgery ended up being. So here you have an electrode on the left hemisphere. Here on the right hemisphere, again, there's two visual cortices. And then there's a third electrode on the frontal cortex. And this is what we call the reference or the ground electrode. It's a region that we're not interested in. And in our experimental manipulation should serve as a reference point that should not change in activity when we do our manipulation. So in this case, the animal is head fixed, so he cannot move, he or she cannot move. And what we're gonna do is we're gonna show something in a screen. Again, we have access to the visual cortex. Anything that the animal sees through the eye, we should see any type of difference in activity. And this is a short video where the animal is sitting around waiting for a response that is going to happen in the screen, and then we are recording the activity before and after the stimulus, which in this case we call familiar because we have shown it to the animal multiple times. That pattern of those bars, black and white bars that you saw, are called phase reversing gratings, and we use them because they have different angles, and we can change the angles and introduce novelty or familiarity to the animals. So what we do, again, going back to the plasticity part, is to then introduce the electrode here into layer 4 of visual cortex. That's not important, but it's a specific layer that is the recipient of the information from the eyes. And then what happens is, in mice specifically, what you get is from the contralateral eye. If we're looking at the right side, and this is the left eye, we receive many more connections from the left eye than from the right eye or ipsilateral eye. How do we know that? Because what we do is we have the monitor. Again, it's a diagram showing you what I showed you in the past. We cover with a patch the ipsilateral eye, get the response of what the animals see in the brain, in the visual cortex. And then what we do is we move that patch to the other side, we record the activity from the ipsi side, and what we observe is something like this. The activity that we observe in the contra eye is much bigger, sometimes from 2 to 3 times bigger than the ipsilateral eye. Again, a lot of research have been done in this area, and we know there's more connections coming from that left eye, so that's why we get more activity. So it's a reflection of just the structural way that this area is developed. So here it's our experimental paradigm that I'm going to show you, and I'm going to try to convince you that we find ways to manipulate it. In juvenile mice, we have this normal initial state. Animals are 21 to around 40 to 60 days old. And we see this normal contraceptive ratio 2:1. If we close the contralateral eye, let's say this is the left eye and we're looking at the right hemisphere electrode, we close this eye for 3 days, open it again, and then record on the screen again. What we see is that the response from that eye got weaker, meaning that now it's a 1:1 ratio from the IPSI and contra eyes. That's called or termed deprived eye depression, because this eye was deprived and it got depressed or weakened because of that monocular deprivation. So we deprived the animal from experience. The cortex adapted using plasticity mechanisms, and now it's 1:1. But importantly, if you pay attention to my title, I don't study juvenile mice necessarily. I try to understand adult mice. So adult mice, what we know is that if we do the exact same manipulation, 3 days of MD, we don't see that deprived eye depression. So animals with this short-term period of deprivation are not capable of adapting and using the plasticity mechanism as a juvenile mice. Why is that? Well, again, this is not research done by me, but by many others. And what we know is that there are these critical sensitive periods, and they are in many sensory cortices around the brain and other regions, where the animals are born and there's an increase in plasticity, and then with time it declines and it goes down. Mention that it goes down to 0. There's some plasticity still happening, but it's much less compared to that peak of plasticity that they had when they were juvenile. So importantly, people have studied why this is the case. What are the things or the molecules that are driving this break in plasticity? And one of those molecules is called PNNs, or perineural nets. I'm going to explain what they are in a minute, but just see here, the red is showing you when they're present, and you can see that very early on they're not present. There's a lot of plasticity, and then they start building up on top of neurons, and plasticity ends. There's a lot of research again done in this area to know that this is the case, and you can manipulate them. But what are perineural nets? They were described by Camille Golgi in the 19th century. A set of neurokeratin, which impeded the spread of current from cell to cell. In that, when he saw it on the scope, what he observed is like this kind of a net that sits on top of specific neurons. And he hypothesized that impeded the spread of current from cell to cell. Now we know that net or corset that he called, it's composed of a lot of things. So promoting protein, sugars, sulfate protoglycans, a lot of different things that compose that net. But all in all, what they do basically is change the amount of electricity that can travel from cell to cell. Because I'm studying visual cortex, I have to show you that this is present in the visual cortex. Perineural nets can be found, for example, in one specific brain region. So people have done a lot of research and found that perineural nets are found in, this is a slice of a mouse visual cortex. So these green things are specific types of neurons called parvalbumin neurons. And those parvalbumin positive interneurons are neurons that are in charge of inhibiting other cells. So what we know right now is that the PNNs or perineural nets sit on top of this specific type of neurons called PV or parvalbumin positive neurons, and that they are not expressed again in the beginning if you use 2 different types of labels for PNNs, and then with time they start building up on top of those neurons until they reach a plateau in adulthood. So again, going back to my critical period diagram, there's an opening of critical period of plasticity. A lot of plasticity is happening, and then PNNs come around on top of PNNs, and there's a closing of perineural of the critical period. More importantly, I told you this is cool and important to understand, but what I'm very interested in is how do we reopen that? How do we get this lower period of plasticity and bring it back to, maybe not necessarily to the same way as juvenile, but at least to do something to enhance that activity. Why am I interested in that? Well, because as you all know, with age, we have a lot of different things, including neurodegeneration. We have changes in learning and memory. And one of the hypotheses, the central hypothesis of why getting older bring all those things, it's because these critical periods are close and the capacity of plasticity in the brain is lower. Therefore, adapting to stimuli or toxicity, it's just not there. So if we can find avenues to reopen these periods, we can then intervene and induce plasticity in the same manner that juvenile animals do. So here I show you since 2013, there were a lot of different interventions already. This top one, CHABC, was the first paper that found a change, and it's a chondroitinase ABC. It's an enzyme that basically goes into the brain. So they have an injection inside of the visual cortex. They release that enzyme and eats away the pyramidal net. And what they show is that animals that receive that treatment have a increased plasticity. Here you can see that it's lower, but it's an ocular dominance index, so lower is a good thing. So here, normal animals, when you do monocular deprivation, like I showed you, they should not change because they're adult mice. And when you have the MD animals, monocular deprived animals with the treatment, you can see a reduction in the ocular dominant index, suggesting that there's a reopening of plasticity. Now, these and many of the other interventions that I mentioned are very invasive, or they take a long, long time. For example, if I go back a little bit, enrichment and darkness, it takes more than 10 days, for example, in the dark to reopen plasticity in a mouse or rat. Enrichment is very heterogeneous. So how do you enrich the environment to induce plasticity? It's very hard to control. So my question in the project is how to eliminate pyramidal nets in a minimally invasive manner to induce juvenile-like plasticity in adult mice. And again, by the title, you can see that I use pharmacology or drugs or digital therapeutics, which is a fancy term to say that we use sensorial stimulation to manipulate the brain activity and therefore, in this case, cortical plasticity. First thing that I did when I joined the lab is try to find what are the candidates to do this. One of them, literature suggested, was ketamine. Ketamine is an NMDA receptor antagonist, and the name, that doesn't matter for you all. But importantly, what this does is that it induces heightened plasticity. For example, in these animals that I'm showing you here, if we injected ketamine 24 hours prior to sacrification and measuring of plasticity through slide physiology, you can see in the light gray animals that they have increased periods of plasticity. This is a normalized response of a slide physiology experience. Again, not important, but what's important is that there's a difference between the control and the ketamine treated animals, suggesting to me that, and many other experiments, to be honest, that ketamine can be used to enhance plasticity. So just to give you a little bit of background, this is a very convoluted slide, but I just wanted to show you what people have found that ketamine does. And again, they bind to NMDA receptors to inhibit, mostly in inhibitory neurons, the type that I showed you in the previous slides that PNN sit on. But importantly, again, here in 2016, they hypothesized that it increases synaptic plasticity by strengthening connections. And it's being used right now at very low concentrations, sub-anesthetic concentrations, for the treatment of depression. Because what I didn't mention is that ketamine, it's a veterinary anesthetic. So if you use it at a high concentration, it's an anesthetic But if you use a sub-anesthetic concentration, people have found that it can actually help with treatment-resistant depression, which is really, really good news. And the mechanisms are still under study, but there are many things that here, like BDNF, brain-derived neurotrophic factor, and other things that help do that. But in my case, my hypothesis was whether ketamine is sufficient to eliminate perineural nets, because people, you know, focus on these intracellular pathways, but I wanted to see, like, what about that corset that forms around the neurons? So what I did is I collaborated with people in Austria that they actually came up with the idea of using ketamine. Sandra is the PI in IST Austria, Alexandra and Francis, who is my grad student that worked closely with me, also UPR, CPR alumni. And we decided to put this hypothesis to the test. The way we did this is we injected KXA, which is a combination of ketamine, xylazine, and zepromycin. The reason why we have these other two drugs is because when you use ketamine at high concentrations, the animals become very hyperactive, and you need other things to tranquilize the animals. Even though they are anesthetized, their muscles are moving. So you don't want that to happen, and you have two tranquilizers for that not to happen. So we have this KXA cocktail, and what we did is we injected it, and then 4 hours later, sacrificed the animals to look at the WFA, or perineural nets, in the cortex. And this is what we saw. We saw that if we injected once with ketamine, the black represents the amount of perineural nets. We still see a lot compared to the saline-treated animals, so the animals that receive no drug. But when we start repeating the dosing every 2 to 3 days, we start seeing that these black dots, which again represent perineural nets, start to disappear. And by 6 injections, they were almost totally gone. So I can show you here the quantification. After 2 injections of ketamine, there's statistically the reduction in both male and female mice of perineural nets. Now, I told you that my hypothesis is that ketamine is the one that does this, so I have to show you that the other 2 compounds are not doing that. So here we injected only the XA, so the 2 tranquilizers alone, to make sure and rule out that those are not the ones that actually do the function to eliminate the perineural nets. And we saw that after 3 injections of XA, the animals don't look different from a control animal. So again, this is suggesting that ketamine is sufficient to induce removal of perineural nets in both male and female mice. Now, if you pay attention to my titles, this says S1. S1 is a different region that I have been talking about. I've been talking about visual cortex all the time. So this is a different region called the somatosensory cortices. And this is because that other lab that I told you studied that region. So I decided to look at the visual cortex. And in the visual cortex, before I go to the visual cortex, one thing that we also did is to look at how much time after the last injection it took for perineural nets to come back. So what we did is we repeated the same dosing 3 times. This is a 3 times KXA. And then we waited 4 hours. Again, you see the reduction, 3 days, 7 days, or 14 days. And we see that by 7 days, by a week later, perineural nets are back to normal, suggesting again that the injection is not eliminating perineural nets forever. Remember at the beginning that I mentioned that we want to create a dynamic process. We don't want to completely distort the system and eliminate perineural nets because they're important for other functions and homeostasis of the brain. So we looked at that. Now, back to visual cortex. We did the experiment, and this is specifically looking at the visual cortex with 3 injections of a KSA, which is the one that I'm going to be focusing again. And we see a statistically significant reduction in both male and female mice with the ketamine injection. So the question, Carmen mentioned, or Dr. Maldonado mentioned in the beginning, that I like to study in vivo electrophysiology. I showed you all of that physiology in the beginning, how they contralateral and ipsilateral look like. So the main question of this project is whether ketamine is also sufficient, not only to eliminate fetal neural nets, but induce functional changes that open a window for juvenile-like plasticity. And that's where the electrophysiology comes in. So I'm going to show you a little bit of the timeline first. We implant electrodes when animals are adults, P60. Then we wait around 1 or 2 weeks for recovery of the surgery. And then we do the 3 times of injections of either KX or saline. Perform the 3 days of monocular deprivation that I showed you before that is not supposed to cause any changes. And then record multiple times, a day that we open, a day later, and 7 days later. If I show you the saline-treated animals, contralateral eye, again, this blue, this left eye, is supposed to be 2-to-1 ratio. That's real data, males and females. And you can see it looks normal. After 3 days of monocular deprivation, even though you see a slight difference, it's not statistically significant. So there's no change, no statistical difference here in terms of when I manipulated the animals by 3 days of monocular deprivation in the saline-treated animals. Now, when we look at the ketamine-treated animals, now we see that there's a reduction, significant reduction of the contralateral eye, not quite to 1-to-1 like I showed you in the past, the juveniles go 1-to-1, but it is a remarkable reduction. Remember, this is a systemic injection of ketamine, so I'm injecting intraperitoneal. And it's not like the other experiment that I showed you that they're injecting directly in the brain, but systemic. So it's very remarkable that 3 times injections of KS systemic is able to remove neural nets and also induce a functional change. So now neurons are able to adapt to this manipulation of monocular deprivation. The way we typically look at these ratios are ocular dominant index, like I showed you before, or the contra-IPSI ratio. So we take every animal and divide it by the contra and the IPSI ratio. And if you can see here, the animals that did not receive the treatment stay at 2-to-1. Again, contra is 2 times what the IPSI is. But the animals that received the ketamine didn't go all the way back to 1-to-1, but around 1.4, 1.5. Many of them. So again, suggesting that we were able to functionally induce deprived depression and therefore reopen the plasticity. Now, the last part of what I want to show you is how this has happened. How we scientists not only want to show that something works, but how it works. And this is many times the most difficult questions. But we have some insight. And the insight comes from research done by other groups where they have shown that microglia, which is a different type of cell in the brain, the one that I'm going to show you here, it's a cell that it's not like neurons. It doesn't communicate by electricity, but it's you can think about it like the immune cell of the brain. And it does a bunch of functions, surveillance. It's involved in phagocytosis, so it eats away things. And this is what you have to pay attention. It's important for synapse monitoring and pruning. So it also makes sure that if you, let's say, a synapse is too big, it goes and it kind of like make it smaller. So there's a bunch of functions that people have seen for this microglia, but importantly, you can see here that it eats away stuff that are extracellular instead of inside of the cell. So we hypothesized that maybe microglia were activated to eat away neural nets. In other words, that ketamine was somehow activating microglia to eat away the neural nets and therefore open up this functional plasticity. To do this, we repeated some of the experiments, but now we eliminated microglia from the brain. So we give animals for around 1.5 weeks this diet that when they eat it, it takes away or kills the microglia in the brain. When we do that and inject 3 times with KXA, our hypothesis is that we wouldn't see any changes in the WFA or pyramidal nets, and that's exactly what we saw in both males and females. There's no reduction. So we, if microglia were not necessary to do this, then we will see that there was a reduction of this staining. But the way microglia gets activated, so the way that this immune cell becomes activated and moves to the area of usual inflammation, that's what they're known for, it's by detecting changes in the environment. Something has to change in order for microglia to go and do something. So one of those things is that ATP, or the molecule that it uses, the cells uses to fuel or energize the cells, it's the one that changes in the environment of that. It tells the microglia, go here and do your job. So we decided to test whether when we injected ketamine in the presence of clopidogrel, which is a blocker of ATP, so it makes microglia incapable of detecting changes in the environment of ATP, if that also blocks the pyramidal net reduction. And indeed, that's what happened. There's no reduction in the pyramidal nets when clopidogrel is present, meaning that somehow there's a change in ATP in the environment that the microglia detects to go and eat away the pyramidal nets. So my last question was whether I can use, so I'm telling you that microglia gets activated by ATP, and ATP is an energizing molecule in the brain and in the whole body, to be honest. So can we use something else to activate microglia and tell it to go and eat microglia? Why? Because even though ketamine is safe to administer, it's still a drug of recreation. It's called Special K in the streets, and it's a drug. So it has a lot of other non-specific things that we don't want to have. So what about if we use sensory stimulation to activate microglia and therefore induce these changes? So what we did is we injected ketamine and looked at the activity.
Uh-oh. Let's see if it comes back. We lost connection with
Héctor. By light flickering. So here you can see, what? ¿Cómo?
Héctor, so
se frizó un poquito. Sí.
Go back one slide.
Go back.
Okay. Here. So here I'm measuring the activity in the visual cortex. This is the raw data of animals that were recorded after I injected ketamine, I injected the of activity in the visual cortex. And what we decided to do is to use this information to try to mimic this with sensory stimulation. I'm not going to show the data of how we analyze this, but basically we found that there were changes in frequency of stimulation when ketamine was present that we could mimic with light. So here is the experimental paradigm of our last experiment, when we show that if we flicker the light at a specific frequency, for example, 8, 40, or 60 hertz, you can, so the eyes is receiving that information, you can actually make the neurons fire at that specific frequency. And that's where the digital therapeutics come in. So you use sensory stimulation to manipulate the activity of the brain. So when we did this at different frequency, what we found is that specifically at 60 hertz, these are animals that were exposed for 5 days, 2 hours per day to the light flickering at different specific frequencies, there's a reduction in the perineural nets. So again, this is for the first time showing that specific environmental stimulation using light are able to not only activate microglia, which other groups have found, but remove perineural nets and induce functional plasticity. So with that, I'm gonna summarize my work, which is a maturation of PV cells and appearance of perineural nets leads to reduction of heightened plasticity. If you disrupt those components of neural nets by using, for example, enzymatic digestion, open up a window of plasticity of synaptic modification in adult mice, and a repeated ketamine treatment, at least more than two injections, are able to remove neural nets in both S1 or somatosensory cortex and B1 visual cortex and restore juvenile-like plasticity. Microglia are necessary for this to happen, and the light stimulation specifically at 60 Hertz, so 60 times per second, is sufficient to remove neural nets in B1, and that requires microglia. I didn't show that data, but I have also tested that. So future plans is to test whether this light flickering is sufficient to induce juvenile-like plasticity. So those are the experiments that the pandemic halted. But the idea is then now to repeat the same functional experiments that I did with ketamine, but with 60 Hertz stimulation, and show that that is sufficient. And to show that many, perhaps other concentrations of ketamine are also able to induce this type of plasticity, and understand the mechanism, not only that microglia activates it, but what molecules specifically are necessary for this to happen. So with that, I want to thank you all for listening, and I'm open for questions.
Thank you, Héctor. That was a wonderful talk. And I want to stress to the students that what you are seeing today is a presentation of a very promising future colleague in an academic setting in not so distant future. And also to show you that just like Héctor started, just like you as an undergraduate in our Department of Biology, he has achieved his academic dream of studying neuroscience and being as successful as he is now at MIT with the data that he's just shown. So I really want to encourage everyone to see Héctor as a role model for what it could be for all of you in the future. But we have questions for you, Héctor.
Yes.
There's one from Dr. Garcia, the first one, obviously, as always. And he says, how are you labeling PNS? And well, that's the first question. How do you label PNS?
So we have two different labels. One is NeuroCam, that labels specific sugar in the net, and the other one is WFA, which is another marker of another component of the WFA of the paranormal net, and both of them show the same type of reduction. Here I show only WFA, but both of them show the same reduction.
Okay, and he says also, why are the PNNS apparently seen only in certain areas of the brain, and it seems that it's only some neurons?
Yes, so that's a very good question. So it seems like I mentioned in the beginning, it seems to be mostly PV neurons, for bulbous neurons, interneurons, inhibitory neurons, but we also see some different distribution along the different layers of the cortex. That we don't understand why. But what we do know is that in layer 4, which is the thalamic, cortical-thalamic recipient of the visual connection, there's a lot of WFA or PNN neural nets, but we don't understand why there's a difference between regions or layers. So that's a good question.
Garcia, are you okay with that?
Sure.
Good. Okay.
I actually have another question, but I'll let the others go and then go back.
Okay. So there's another one, and this is from Maria Rivera, one of our students. She says, is there a meaningful difference between the efficiency of using ketamine for the elimination of the PNNS across time between males and females?
We saw that females eliminated PNNs much faster. Meaning like they needed less dosing in terms of for eliminating. At the end of the day, they both look at after 2 injections they go lower, but the effect size, so how much they are eliminated after 2 doses is much more with females than males. We don't understand why, but we do see that there's a sex difference that at the end of the day, kind of after 3, that's why we chose 3 injections, because with 3 injections, both of the sexes look the same. And we wanted to use both to do our functional experiments, but we do see some sex differences.
Right. Then there's another question from un tocayo tuyo, Héctor Jado. He says, have you considered looking at the effects of ketamine on plasticity in the hippocampus?
I haven't, but others have. The experiment that I showed you before in rats was specifically in the hippocampus, in the Schaefer collaterals, and it has been shown that it induces higher amounts of plasticity, so LTP, long-term potentiation. So I haven't, but yeah, it has been done. Okay. Now, people don't know if the reason why we see higher LTP in the hippocampus is because of peronal nets removal. That would be a good question to ask, but at this moment, I'm not doing that.
Okay. Then we have another of our students, Cielo, who says, you mentioned clopidogrel, known as Plavix, inhibits the detection mechanisms of microglia, right? And in Alzheimer's and other neurodegenerative diseases, we know so far that proteins build up between cells. Has it been shown whether Plavix effects on microglia is behind this buildup, considering they are not doing their job at cleaning debris within the nervous system?
Great question. No, I'm not aware of a study that looked at that, but yeah, very good question. Using Plavix, like many people use Plavix, is definitely going to eliminate some of the microglia. So it's yeah, I don't know.
Yeah, could be interesting. And so there's another, Victoria asked another student, she says, how fast do PNS come back to normal with the 60 Hertz stimulation therapy?
Ooh, good question. We haven't finished those recovery experiments. The last time that I look at the data, it looks very similar in terms of 1 week. Now we think it comes back a little bit faster than that. So 1 week, they're definitely now again 3 to 5 days. We see also we think it's gonna reach statistical significance. So the light flickering is not as potent as ketamine. That's something that I have to mention for sure. It's a sensorial stimulation, and it doesn't reduce peronal nets the same effect size we saw with ketamine. So we expected that they come back faster, and it seems like they do.
Great. There's another question from Maria Rivera. She says, could it be possible for other stimuli, for example, sound, be used to induce PNN removal in the areas outside the visual cortex?
Great question. Yes. I haven't tested it, but I know of a lab here at MIT Wayside. She uses light and sound to induce gamma oscillations, so gamma between 30 to 80 Hertz stimulation. And she has shown that induces microglia activation. From there to PNN removal, she hasn't done those experiments because she's interested in other things, but it is a good hypothesis. So yes, sound can be used. The thing about sound is that it's gonna impact different brain regions, of course, auditory cortex and other regions. So using multimodal sensorial stimulation can definitely impact different brain regions.
Okay. Then Paola Morales asks, how did you identify ketamine in the PNN?
No, we didn't identify ketamine in the PNNs. We just injected ketamine to test the hypothesis of removal.
Okay. And then another question. So are you using rats for a specific reason? I ask because as far as I know, zebrafish have been shown to be great models for synaptic plasticity, right?
I use mice, not rats, but yeah, the reason why I use mice, it's twofold. First of all, most of the research in the visual cortex field have been done in cats and mice, and we have a lot of the genetic tools to manipulate the system. But yeah, of course, zebrafish is another great model. C. elegans, for example, also is another good model. But the thing about those animals is that behaviorally, I don't have the tools to do it in my. I focus on mice where we know exactly what's going to happen, and then I can manipulate those variables. But yeah, it could be done in several features.
Okay, and then the last question from one of our students says, so if they come back faster, is it possible this therapy requires dosage to be continually increased for it to function?
Good question. I have to answer that question by first telling you, like showing you that it is sufficient, that 5 days of stimulation is sufficient to induce functional plasticity. I do have data on that. It's not complete, but I think it does. And after that, I have to show you that it's maintained throughout time. So I don't know the answer yet, but yeah, it could be that it requires not necessarily more stimulation in terms of the frequency, but at least many more times than ketamine.
Great. And Dr. Garcia.
Okay, so Héctor,
Yep.
let's talk about true plasticity. So you give them ketamine and you reduce the dominant eye activity. What if you then patch up the dominant eye? Does the other eye takes over? And is it permanent?
I don't think it's permanent. I started backwards, but I don't think it's permanent because of our hypothesis that PNNs, when they come back, it takes over. But I do think that if we patch the good eye, or the dominant eye, the other eye takes over. We do think that. We haven't done the experiment, but we have a lot of evidence from other experiments in the lab using other drugs that that's going to happen.
Thank you.
Okay. Well, I think we have finished all the questions. As you can see, you've been grilled by our students.
I love it. I love it.
And it's just an example of the potential that we still have in our institution and in our community at the BP-Endure. And so we are very proud of that. And we thank you, Héctor, for being here with us today. So now we continue toward the recruitment component of our seminar, which is with Dr. Sansafar. And there she is. Wonderful. She's here. So you're still muted, Mandana, so when you get a chance. So we could hear you. Nope, not yet.
Okay, can you hear me now?
Now we can.
All right.
Okay, great. So take it away.
Thank you. I just want to check first, students, are you all undergrads, seniors, grad students? What do we have in there?
Okay, well, we have a mix, I think. I don't know. We have sophomores. I know junior sophomores.
Okay.
And several seniors that are, already applying for graduate school.
Okay.
And others are considering post-bacs.
All right.
I have a couple there that I know they're looking for a post-bac.
Okay.
And summer training for sure.
All right. So let me try to share my screen.
Yes,
I usually don't do Google. So
we have a freshman. We have a freshman. A freshman.
Yeah, that's okay. So how do I share my screen on this Google thing?
Okay, you have to go to, on the right-hand side, there's going to say present now.
If you go right-hand side, top or bottom?
Bottom.
Right-hand side, present now. Yeah, I see it. Okay.
You click there and you say...
your entire screen?
Yes, you can do that.
Okay. Hello. All
Here
right.
we are. We're going to see all your screen.
Okay. Do you see my screen now?
Yes.
Okay.
And then, yeah. So you can do slideshow. There you go. We're perfect.
All right.
Okay.
And I don't see myself. I guess that's fine. And I don't see anyone. All right.
Yes.
So I'll try to. All right. So I'm here to talk to you about a few programs, but I always like to. It's great to come to Puerto Rico. This is a slide that I took, I think, 3 or 4 years ago. Here is Héctor. And these are all students and postdocs, graduate students and postdocs who were from UPR. And so here, Emir and Nicole and Monica and Cassandra actually were from UPR, Rio Piedras. And I think, I don't know if Nicole was in BP Andur, but Monica was in BP Andur. And in fact, she worked with Dr. Garcia Raras. And so all of these guys have graduated. Actually, Monica and Emir and Nicole all graduated this year. And then we have here Francis, who is actually, I don't know if he was in BP Andur, he was in Mark, and he's actually working in the cell lab as Héctor. And then we have Nancy, I think, Carmen, you know Nancy, right? Nancy Padilla?
Yes, of course.
Yeah, she was in our summer program, and then she went to Colombia to do her PhD, and then she came back to be a postdoc with us, and it was great. And now she's in San Diego because her PI moved. But anyway, this is to just show you that we have very happy Puerto Rican community.
Yes, I meant to tell you that Nancy just got the L'Oreal.
Oh, really? Yeah.
Yes.
Oh, she's great. I'm so happy for her. That's a big fellowship. Yeah, that's great. Yeah. All right. And then, so I represent several departments. I mean, I'm a biologist, biochemist by training, but I also do the program for brain and cognitive science and computational and systems biology. And I'm going to talk to you about all three of these programs. So the first one is a quantitative methods workshop that every year BP Andur sends students to our campus, and they work together. They work for 6 days straight to learn MATLAB. And unfortunately, this year it's going to be virtual, but we already have 75 students signed up, and the deadline is, I think, tomorrow. So if you guys are interested, talk to Dr. Garcia and Dr. Maldonado, and they will finalize the list. But Carmen, you sent me four names already, right? So these are your final list?
Yes.
I think? Yes.
Thus far, yes, I think so. Yeah.
Excellent. So, so you guys will receive a link, and it's very important that you register on time. And just to go through what that quantitative method workshop is, we only invite 8 schools, and we always invite the UPR students to come into our program. This year it's going to be January 4th to 9, but again, entirely virtual. There are going to be faculty lecture every day, then a small group discussion with faculty, and then 4 hours of computer lab every day. And it's important, it's full-time, so it's important that whoever comes to that workshop does not plan anything else, like not taking courses, not, no part-time job or anything like this. And so, for example, this was last year, students from Puerto Rico were having a long discussion with Eliezer Callo. And Eliezer Callo was a Mark student at UPR in chemistry. In 2000, he graduated in 2006 from UPR. And then 4 years ago, he started as a faculty in biology in our department, and it's great. And actually here we see Paola. Paola was in our summer program, and she actually was working directly with Héctor on a computer, on a thing. So this was some of last year, and this is the contingent we had from Puerto Rico. So we usually take 75 students every year, and always a third of the students come from Puerto Rico. So this side were all the students from Rio Piedras, and then in the middle we had the students from Mayagüez, and here we had the students from Humacao, and some of those ended up in our summer program in the summer. So we can only take 6 or 7 students per campus. So every year we have students who are unable to take our course. Now this course, part of it is online, and it's offered twice or 3 times a year. It's 8 weeks course, and the next start date is February 9th. So if you have never had any coding experience, I strongly encourage you to take this course. You are going to have an idea of what R is, what MATLAB is, and what Python is. It's about 5 or 6 hours a week of work. And if you want to get a certificate, it costs $99. And what some of the schools have started to do, I mean, the RISE program at Mayagüez started to do this, is to actually cover the cost of the certificate for the students, because you can only get a certificate if you complete 80% of the work. And that's the only way for you to learn. So I strongly encourage anyone who is interested in how to use coding in data analysis in biology and neuroscience to take this course. Any question about the workshop before I switch to the summer internship? I cannot see anyone. So is there anyone having a question? No? So just to remind you, you have to go through Dr. Garcia Raras and Dr. Maldonado to sign up with us. For the summer research internship, this is great. I mean, we have had a lot of students coming to the summer from Puerto Rico. It's 10 weeks. The deadline is January 30th, and the website is, the online application is actually open right now. So you can go to that website and learn about the program and apply. And like most of the other program, there's a stipend, on-campus housing. We are assuming that it's not going to be virtual. So we had a virtual summer program this year, but it's not going to be, at this point, it's not going to be virtual for the summer of 21. And what's very important is the level, the quality of the mentorship for our students, and also the community building that we have through our students. So, so the requirement, we need the GPA of 3.5 or more. You have to have prior research experience to come to this program, and we require 3 letters of references, especially one, or at least one, if not 2, from research mentors. And I don't know how many of you had any research experience this past summer. We understand the issues. We know that some students had their summer internship canceled, but regardless, do not let this deter you, because we know a lot of students are nervous about this, but everybody was in the same boat. And again, these are some examples. So here we have Gabriela. She came to our summer program twice, and she's now a first-year graduate student in biology. This is Raul. He also came twice, and he's actually in the brain and cognitive science department now. And here's Nicole, who actually probably mentored him while he was still an undergrad. So he's now starting his 2nd year in the PhD program in brain and cognitive science, and he's working in systems neuroscience. And this is the students we had from Puerto Rico this past summer in our virtual summer. And we have these 3 students were from Mayagüez, and these 2 were from Humacao. And what is unfortunate is we didn't get anyone from Rio Piedras, and that's a pity, and I'd like this to change a little bit. These are some of our students from the MARC program in the past. Isaris here is now working in a biotechnology lab. She came to our summer program and then came to our graduate program. This is Nancy when she was in our summer program. When was it? I don't know, 10 years ago. She hasn't changed a bit.
Yes.
And her mentor at the time was a grad student, and now he's a faculty at Howard in neuroscience. And here we have Yamil, who is also in biotech. And I forgot Dr. Garcia. What's his name? You remember? You're one of your best students?
I think Jose
Enrique, that's right, Enrique. Yeah, Enrique was in our summer program, and then he spent a gap year also being a technician in one of the lab, and then went to Harvard for his PhD, and now I think he's in patent law or something. So I like to show this. And just for the history, so I have been coming to Puerto Rico since 2004, I know, I think. And up to now, we have had 85 students from UTPR in our program, in our summer program, and 26 of these students came to MIT for grad school, and 12 have already graduated with PhD, 3 with masters, and we have 9 current who are PhD students. And again, it goes between having people. So here is our, the first 7 students or whatever, 8 students that we had from Puerto Rico. They have all graduated. He's now a faculty at Columbia, and Eliezer is a faculty at MIT. So Brian Souza here and Isaris were from Rio Piedras. All the others were from Mayagüez, and Eliezer was also from Rio Piedras. And so they're very, very good. And Francisco and Yadira are both applying to be faculty. They are currently doing the postdoc in New York in cancer. And Francisco has gotten the Hannah Gray, HHMI Hannah Gray, which is a very prestigious postdoctoral fellowship. And then these are some of our other students, not all of them, but again, these 4 right here have just graduated all this year. And Monica again, and Nicole and Emir were all in Rio Piedras. And these 3 students are from Mayagüez. But again, what's happening is we're starting to get students from Mayagüez and Humacao in our graduate program. And I'm trying to, again, see what is, deterring you, if it's, Héctor is here, so he can talk to you about the culture at MIT. It's very supportive, I think. And there is a lot of, it's not cutthroat. I don't know what is your idea of MIT, but unless you come to the summer program or the workshop, you don't really, experience what is the culture. And every year we go to Abrahams, and this was, I think, two 2 years or 3 years ago. So Francis was presenting as an undergrad what he had done at MIT, and Nicole was actually manning the booth. Héctor was judging, Eliezer was there as a faculty, and then you probably all know Orestes Quesada. He was also there, as part of a program director for Mark. And this was last year, 2019. We were all at Abracam. This year it was all, in virtual, but last year, these were all of the students from our summer program throughout the years were at Abracam. So some of them, like Eliezer, were faculty, some of them were presenting as undergrad, some of them were representing other schools. So we have a student here from Berkeley who is from Mayagüez, and we have another student, I think she was Lydiette from Rio Piedras, who is now at UT Southwestern, and then Francis is here, and so on. So we are very involved trying to get students to come to our summer program, and usually when they come to our summer program, they love MIT, and then we try to get them in there. Now, we also have a post-bac program. This is specifically for brain and cognitive science. It's a 2-year post-bac program. It's actually unusual because you actually get to take courses for credit officially at MIT. So the program covers your tuition and it covers your stipend, which is equivalent to a stipend of a grad student. So it's a very generous stipend compared to all other post-bac program. It's around $39,000. And these are some of our past post-bac students, all in brain and cognitive science, who are now in grad school. And this was one of our first post-bac student in 2014, and he's now finishing his 5th year in neuroscience at Stanford, and he just published a huge paper in neuroscience. And I just want to mention Josefina, who was from UPR Rio Piedras. She came to our, I met with her when I visited, the campus. Then she came to our workshop, quantitative workshop. Then she applied and got admitted to our summer program, and then she applied to our post-bac program and got admitted to our post-bac program, which is a 2-year program. But she progressed so fast that after 1 year, we admitted her to our PhD program, and now she's starting her 2nd year, and she's now doing a lot of statistics and mathematics, doing modeling of the brain under anesthesia. All right. So some of you, I don't know how many of you are graduating in May and what you want to do. If you don't want to go straight to grad school, I want to remind you that post-bac is not the only way to get some kind of practical training or whatever you think you need in order to go to grad school. And for example, Enrique, who was a very strong student, was not in a post-bac program. He was hired as a technician in one of the lab at MIT, and then went on to go to Harvard. I mean, he was admitted both at Harvard and MIT and every place he applied. So I just want to remind you that post-bac is not the only way to get training, and sometimes a technician job is actually better than a post-bac. It just depends on whose lab you get in and if you get the same attention. So I'd like people to think about this. I also want to talk about the PhD program. As of this coming year, no GRE requirement, not at MIT and probably not in many other programs. So please do not spend a dime on a GRE prep course. Do not spend a minute trying to do a GRE. Spend time, doing research in a lab or reading papers. That's going to be much more important for you to do your work. And these are some of our faculty. What is important is you are fully funded for the entire time where we are in our program. You get, mentored by caring faculty. This is Nancy Canwisher. She's a great mentor and speaker, and I think she actually spoke once at one of the neuroscience meetings at UPR.
Yes, she did.
Yeah, and we have a strong supportive community. Héctor can talk to you also about the diversity and equity efforts that the Brain and Cognitive Science Department is doing. But this is across MIT, and again, I recruit for 2 programs, but I am a biologist by training. And right now, how much time do I have?
Well, you still have 15 minutes, so maybe.
Okay. So I'll take some question before, and then I want to talk to you about one type of research that is going to change a lot because of what was in the news recently. But do you have any questions before I go forward?
There's a question. There's a student that is not part of the BP Endure Rio Piedras, but she's from another BP Endure.
Right.
And she says, could she also apply for the workshop on the?
Again, it's up to you because you give us the names, right? So we do not select. So the selection is done by the faculty. So if she's part of the MBPNDR, you guys decide, if she can, depending on how many slots are left, if she's, if she should come. Yeah.
Okay.
So we don't make that decision. We decide for the post-bac, we decide for the summer program, but I leave this to the schools to choose the students.
Okay. Maria, which university are you affiliated to right now? If you can unmute yourself, if you're still here. Let's see her. But she left that question. See? Maria Rivera. Yes, Maria. Yes? You were gonna say? We don't hear you. Oh, she seems to have problem with the.
Maybe she can type. Can you type in the chat?
It's at WashU. She's at WashU.
Oh, WashU. Oh, you guys have a program with WashU?
Yeah.
Oh, well, it depends. I mean, we don't take students from WashU. We take students from UPR. So is she at WashU or is she at UPR?
No, she's at WashU because she's the Endure community from other partnering institutions.
Right. No, so if she's not in, because we partner with institutions. Yes, so we partner with the BP Endure at NYU or Hunter, but that's pretty much the other one. Yeah, the only other one, BP Endure. So no, so if you are not from one of our partner institution, yes.
She was a former UPR student, but she's now at WashU. That's what she wants.
Right, yeah. No, unfortunately, no, especially that we have already reached our maximum. I mean,
Okay.
Yeah.
But that's the only question we have right now. We want to mention that if we are putting available like a document, Google Doc for if they want to have a one-on-one interaction with you, Mandana. I just click on, I gave them the how you say, the link.
The link. So is this the one that Patty Ordonez organized?
Yes, that's her.
Okay, yeah. So if you want one-on-one to talk about the summer program, graduate program, postdoc, applying to technician position, I'm available tomorrow. So I think I have 3 or 4 slots available. I'll be happy.
The link is there. Mm-hmm. Any questions for Dr. Sansamafar before she continues to, or any things about, environment at MIT, living in Boston or Cambridge, which is lovely.
And again, I can refer you to grad students, who are either, actually I could even send you a couple links. We just had the news on Amaris. Amaris is here, and she after she graduated from MIT, she went to work for Vertex for 3 or 4 years, and right now she moved to Amgen, and so she's right, walking distance to the MIT campus, and she stopped to talk to some of our summer students. So I can, and so she never thought she could live in Boston, and now she's married and living in Boston. So and then Héctor can tell you what's how it is to live in Boston, the winters, the summers, and so, because that all matters. I mean, the summer it's easy, but when you choose a grad program, you have to make sure you are comfortable, because it's going to be 4 or 5 years before you graduate. All right, any questions?
Yes. Is there a MSRP bio? It has in the MSRP bio, it has faculty of the biology department, or?
So in the MSRP bio program, it's all of the faculty in biology, all of the faculty in brain and cognitive science, and this year we're also getting a few faculty from chemistry. So
who's asking Paola? She's doing bioengineering. That's her undergraduate degree. Right. So her question is, would a person with a background in engineering will fit in this kind of program?
Oh, definitely. Yeah, I mean, we definitely have people from bioengineering coming to biology. Yeah, all you need is to have, to have some research experience and have taken, a strong background in biology, and then bioengineering, of course, that's not going to be an issue. We have 5 of our faculty are cross-listed in bioengineering also. Okay. So in fact, Amy Keating, who is head of the graduate program, is also on the committee for application for bioengineering. So there's a lot of crosstalk between departments. Yes.
Okay.
Other questions?
Don't see.
No.
They're
Okay.
kind of shy. You can unmute yourselves if you want to ask
Yeah.
a direct question to
All
Dr.
right.
Sansafar.
So I'm going to move then to something else here.
Uh-huh.
But then usually I call students. So Carmen, you need to ask to help me. Just randomly call on student because I cannot see their
Okay.
faces.
Sure,
Okay.
sure.
So I want somebody, just pick someone to tell me what this is.
Okay. Viviana Valentin.
Yeah, Viviana, unmute yourself. What do you think this is?
¿Qué es
Okay,
eso?
so I'm seeing like the structure of a protein and
Okay.
possibly a ligand in the middle of the picture.
Okay, excellent. Can you tell me a little more about this protein?
I mean, I guess I'm seeing the helixes
Yes,
and
excellent.
the
Yes,
plates,
yes.
but I don't know. I think that yellow is the point of contact with the ligand, but I don't know what else could I say
Okay.
about this.
And you need to tell one more thing about this protein. So yes, they are helices, they are beta strands. So here we're looking at, you know, both the secondary structure and the tertiary structure. Something else about that protein? One more word that could add a little more detail. Or anyone else, Carmen, you can pick someone else. So I need one more detail about this protein.
Okay. Claudia Irizarry. A ver, Claudia. Let's see. She's Claudia. Don't be shy.
Yeah, I can see you, so don't be shy.
And what about Héctor Haddock? Vamos a ver, Héctor. Héctor.
And does it have two subunits?
Yes, that's the other thing I wanted to know. So here, and this is a beautiful movie, you know, showing this. So you can see here the beta strand, beautiful here in the center, and then you have 3 helices here. You have a loop here. You have this other subunit. So you have 2 subunits. So this is a heterodimer because obviously the 2 subunits are different. And here is the field model that is showing you actually the interface, the contact between the two heterodimer. And what's important here is that, I don't know if you guys know, but in lot of cancers, there are some proteins that are active and then form heterodimers. So one of the idea of having cancer drugs is to break that interface between protein. And the point here is to show you how difficult it is, because this is a lot of contact, and people usually are hoping to get a peptide to disrupt the protein-protein interaction, and it's going to be very different. And this is actually the SRP receptor, and this was the GTP. Now, the next question is, so this is a beautiful model. What do you think it took to actually find the structure of this protein? Okay, so because right now we are giving you the example. So you see the secondary structure, and then you actually see how the atoms are put together. So this is the 3D structure of a receptor. My question is, what type of experiments do you have to do to get this kind of experience or this kind of information? Carmen, just pick someone else.
Ian Diaz. A ver, Ian, what do you think you need to do?
I'm not very confident in my molecular knowledge to answer that kind of question, but thanks for picking my name out of the lot.
But can you guess something? Just try. What do you think? What type of experiment do people have to do to find out the shape of a protein in space, the 3D structure of a protein, and where each of the amino acids sits and how do they interact with each other? What is your major?
I don't know, but I'm gonna go ahead and read from the chat. Somebody said crystallography.
Yes, exactly, crystallography. Okay, so what does it take to do crystallography?
Maria from WashU is answering.
Maria, what is it?
A ver, Maria.
So what do you need to do?
Problem with her mic, that's why she can't type. So she says it's crystallography.
Yes.
But,
yes, you need crystallography. Excellent. So here, beautiful crystal. But my question is, what did you need to get? I mean, what are these crystals made of? I mean, how do you get these crystals?
Okay. Any guesses before I start calling names? People are leaving, Mandana. They got scared. But
even if I cannot see them?
Well, you can, yeah, I don't know. Let's see, Luisa. Let's see, Luisa. Let's see.
Oh my God.
Or anyone, tap in the chat. Maybe you guys can tap in the chat. So how do you get crystals, protein crystals? Because that's what you need to then do diffraction with X-ray and then get the pattern.
Maria answers, Rivera, concentrate the protein by evaporating the solvents.
Yes, and how do you get these proteins? That's true. You concentrate the proteins, and what can you tell me about how you prepare the protein?
How do you prepare it, Maria?
Or what do you need to have? What kind of protein do you need to have to make these crystals?
Any other people?
You need very pure protein. Not only, you need a lot of very pure protein. So this is just 2 subunits I showed you. So here are the E. coli extracts. So they are highly expressed because they have been cloned. So here is one of the subunits. What is the other one? And eventually they are purified. This is not good enough, okay, because you have a lot of other stuff in there. And then this is another level of purification. So I hope you see how much of these proteins you have, both of them. Okay, this is the peak, but this is still not good enough. And there is another step that I'm not showing here where that band is left. So now you have very concentrated and extremely pure protein. And now you can hope to crystallize them. And then if you crystallize them, then you hope that you can go, and X-ray them and they won't break. And then you will get a nice pattern and so on. And I don't know if you guys saw in the news, this was in the news yesterday. And in fact, it is world news. For the last 50 years, people have tried, biochemists have tried to figure out from the linear sequence of amino acid, what's going to be the structure, the 3D structure of protein. And why is this important? Because proteins only work when they're correctly folded. So people have tried, and this took a lot of computer power, for supercomputer power to try to predict how a protein fold, and they were able to do it maybe at best for 50 amino acids, because you have so much, you have to look at minimal energization calculation. And one of the company in England, which is DeepMind, worked on this for literally 2 years, and they literally are now able to find the 3D structure, predict the 3D structure of a protein based on its primary structure. Okay, so this might revolutionize, science, because I don't think people are now going to crystallize protein as much, because that up to now was one of the only way to really figure out how a protein is folded, and then you can start doing site-directed mutagenesis. It's important for neuroscience. It's important for any kind of protein you study, and now this is a breakthrough. So I wanted to talk to you about this, and this was entirely done with artificial intelligence, and I think probably 10 computer scientists and 1 biologist probably to do this. All right, now I could talk more about science, but I chose one thing that is really a breakthrough in science, because they have been working on it for 40 years. So now, how do I unshare my screen?
You just do end sharing.
Oh, I could show you this too. Yeah. Okay. Who can tell me what this is? Anyone?
The size of a mouse brain.
Yes, it's a slice of a mouse brain, and I want you to see what happens here to that mouse brain. Okay.
Mm-hmm.
So how many people are left there?
We have 18.
18. So pick someone who can tell me what you think you see, and you don't have to, tell me everything. Just, again, it's just an observation. Okay. What do you think is happening?
Paola, dile que tú ves ahí a la doctora Sansamar. Paola. What do you see? What happens to the tissue?
Is it enlarging? Is it absorbing the fluid?
Yes, it is enlarging. Now, what's the interest in enlarging this tissue?
It was really little, and now it became big.
Okay, so this was the original size, and now this is the next slide. So what's the interest? Why would you want to do that?
You can do imaging on it.
Yes.
And you can also observe the structures in more detail.
Exactly. So you made it bigger, right? But for this to be useful, you know, how do you want this tissue to increase in size? So you go from smaller to bigger, but what's important in that process?
Proportionally?
Yes. Okay, that's the key, because if you start, so this tissue is swelling. So if it's starting to swell, you know, on one side and not the other, or parts of the tissue are going to swell faster than others, you are going to lose that original structure. So this is actually, anyone knows what this process is called here? It was devised at MIT. It's called expansion microscopy. Okay, and it's done in Ed Boyden's lab. And Ed Boyden is a neuroengineer, and he likes to find techniques like optogenetics and others to actually help neuroscience move forward. So here the idea was to expand the tissue so you can look at the details of the tissue without having an expensive microscope. So the idea is now, instead of having a 100x, you can look at this tissue with a 20x. But the key thing, of course, is you are swelling this tissue proportionally. So of course you are going to have to treat the tissue to remove anything that is going to prevent the swelling. And actually the chemical that they are using for the swelling is the same chemical that they are using, or polymer that they are using in diapers. So I don't know if you guys have changed babies' diapers, but you get that, you know, you absorb a lot of the liquid in the diaper. And so that was one of the polymers they used. So I just wanted to show you this. And now I'm still trying to see how I can get out of here. So where do I
stop? Do you stop sharing? There's a blue button in the middle that should say stop sharing.
In the middle of
your screen. Yeah, here in the chat.
In the chat.
I don't see the stop share. Oh, here we go.
You are presenting. So right now it says you are presenting. So
yeah,
how do I stop presenting?
Okay. Yes.
All right. Okay. So I hope you I don't know how many of them answered questions, Carmen.
Oh, well, yeah, you had some questions, but I think they're all going to connect to personally, maybe tomorrow for one-on-one.
Okay, yeah.
And then,
so, yeah. And don't be scared of me. You know, I might look scary, but I don't bite.
No.
You know, don't be scared, and come and ask me about any question you want, and I can give you advice. You know, free advice that you can take or not. You know, about, you know, what's the best course for you, for the goals you have in your, you know, what career goals you have. And and I hope that we start building more, you know, Hector, you also have to help recruit for MIT. I don't know. I mean, you know, I get too many students from Mayaguez and Humacao, and we had one student from Calle, in fact, and we had one student from Arecibo. I think, what happened? The thing collapsed?
No, yes, they did. It did, unfortunately.
Yeah.
A major, a national tragedy for
Puerto. No, I saw the picture. Yeah.
But now we are at the end of our time. I really want to appreciate Héctor and, of course, Dr. Sansafar for being here. And so you know that next week we have another seminar from Dr. Edith Brignoni, who also was a Neuro-ID student, but now she's a postdoc at Stanford University. So now we had Héctor and then Edith. And therefore, we are looking forward to also listening to what she has done for the last couple years at Stanford. But thank you so much for being here, and I hope all our students consider MIT as of course, as a chance for growth and development as a neuroscientist. Thank you.
Thank you. And okay.
And